Every time you pick up a knife to chop an onion, you are initiating the real-time manufacture of a literal airborne chemical weapon. The stinging in your eyes is a biological defense system designed to burn the face of any creature that tries to eat it. Humans consume about 20 pounds of onions per person every year. We treat the bulb as a culinary staple, even though the plant is a sophisticated delivery system for an airborne irritant that creates a microscopic drop of acid right on your cornea. Crying over a cutting board is the successful deployment of a plant's biological defense system. It is the result of millions of years of evolution designed to discourage anything from taking a second bite. To stop those tears, home cooks often rely on traditional tricks passed down through generations. People suggest lighting a candle, chewing a piece of mint gum, or biting a slice of bread. These methods fail because they ignore the chemistry at the source. A small candle flame cannot consume volatile sulfur compounds fast enough to protect your eyes, and biting a piece of bread only changes how you breathe. Effectively stopping the crying requires treating the kitchen counter like a laboratory. We have to track the exact molecular sequence that is triggered the millisecond that metal blade hits the plant tissue. By mapping this precise biochemical cascade, we can identify specific vulnerabilities in the onion's defense and use that data to biologically disarm the weapon. Onions evolved over millennia alongside hungry soil predators. They developed a sophisticated chemical deterrent that stays safe until physically damaged. Within an intact onion cell, the ingredients for this defense are stored in completely different compartments. One side holds a class of sulfur-containing amino acids, the fuel. On the other side, locked away in a separate vacuole, is an enzyme called alliinase, the trigger. When you slice the onion, your knife causes the cell walls to rupture. The internal barriers break down, and the fuel and the trigger enzyme are forced to mix for the first time. Within milliseconds, the alliinase rips apart the amino acids. This creates a cocktail of sulfenic acids, pyruvic acid, and trace amounts of ammonia, the compounds responsible for the onion's signature pungent smell and tangy flavor. At this stage of the reaction, the onion is flavorful and aromatic, but it is still entirely harmless to the human eye. Food chemists once assumed the tear-causing molecule formed by accident. However, in 2002, researchers discovered a second, highly specialized enzyme inside the onion that exists for the sole purpose of manufacturing an irritant. This second enzyme is called lachrymatory factor synthase, or LFS. It catches the newly formed sulfenic acids, twists their molecular structure, and releases them as a highly volatile gas called syn-propanethial-S-oxide. In 2017, crystallographers at Case Western Reserve University finally solved the 3D structure of this enzyme. Research showed the onion repurposed an ancient protein scaffold into a highly specialized chemical weapon. This gas is exquisitely volatile. It boils off the cut surface of the onion, drifts into the air, and dissolves into your tear film. Once touching your eye, the gas reacts with water to form trace concentrations of sulfuric acid. Immediately, your corneous nerve endings detect the chemical irritation and trigger the lacrimal glands to flood the eye with protective fluid. The resulting tears are an emergency anatomical rinse cycle initiated by the LFS enzyme pathway. This makes the LFS enzyme the primary target for any effective kitchen strategy. By mapping the molecular steps of this attack, we can identify 3 specific ways to sabotage the enzymes in the fuel before the gas ever reaches your face. The first tactic is chilling the onion. Enzymes are microscopic protein machines that work fastest at warm temperatures. Dropping the temperature makes both alliinase and LFS sluggish and inefficient. Research from the University of New South Wales found that refrigerating an onion for 30 minutes before cutting reduces the total volume of emitted gas by approximately 60%. By leaving the root intact on the board, you sequester the vast majority of the precursor fuel. You keep it locked away from the reaction until the final seconds of your prep. Modern science has even produced varieties like Sunions, crossbred to have naturally low-activity LFS enzymes, significantly reducing the amount of gas they produce. Understanding the molecular mechanism of your ingredients allows you to stop fighting your food and start controlling it. When you know how the weapon works, you finally know how to disarm it.